Hello everyone. Now we will start the lecture second for the medicinal chemistry first, that is the physicochemical properties. First point, introduction of the physicochemical properties. The biological response of a drug molecule will come when the drug reaches in a sufficient concentration at the site of action and it interacts with the receptors. Means we will get the biological response of a drug molecule when a drug reaches at the site of action and it interacts with the enzymes. Then we will obtain its biological response. So in order to do this, a drug molecule has to fit the receptor. The physicochemical properties and the stereochemical features of a drug molecule affect the transport of the drug molecule from the absorption to its site of action. The various physicochemical parameters affecting the drug actions are ionization, solubility, partition coefficient, hydrogen bonding, protein binding, chelation, and bioisosterisms. For example, see, the pK value, that is the dissociation constant, is the parameter which indicates the strength of the drugs in terms of its acid-base reaction in water. It can be seen that the drug having dissociation constant in the range of 2 to 4, they get readily absorbed by the stomach, and because that they remain largely unionized in the acidic media of the stomach. Means in this way the each and every physicochemical parameters that affects the human body, so it is important to know the various properties of such physicochemical parameters. In that, the first is the ionization. So for a good solubility and the absorption of the drug, it is important to maintain the balance of ionized and unionized form of the drug. When we should have the solubility and absorption of the drug, so we should maintain the balance of ionization and non-ionized form of drugs. This is essential because the ionized form have good water solubility and hence the good binding capacity with its receptors. The non-ionized forms helps the drug to cross the cell membrane. Since the most of the drugs are either weak acids or bases, so their absorption from the gastrointestinal tract take place in unionized form. The unionized form is a function of both dissociation constant and pH of the environment. So for the acid, the value of the pH is shown in this equation, and for the base, the value or the equation of the pH with the dissociation constant of the base is given in the equation. Now moving toward the next point, the weak or very weak acidic drugs are mostly absorbed in the gastrointestinal tract due to the presence of microvilli and the large surface area as compared to the stomach. But the basic drugs, they will also absorb in the intestinal tract where the pH is around the 8. Some drugs occur in unionized form and poorly absorbed due to their low lipid solubility. In some compounds, minor changes can lead to the significant change in the ionization, and further it leads to the differences in the biological activity. For example, the barbituric acid and its 5 monosubstituted derivatives. These are inactive and lack the CNS activity, while its 5,5 disubstituted derivatives can having the activity of CNS depressant. Now the next parameter, that is a solubility. The solubility of a drug in a polar solvents and a non-polar solvents depends upon various factors like chemical structure, particle size, and crystal form. The polar non-ionic compounds form hydrogen bonds with water whereas non-polar compounds form hydrophobic bonds with the lipid and they get dispersed. Generally, in homologous series, lipid-soluble drug they possess high biological activity as compared to the water-soluble drugs. For example, phenobarbital is insoluble in water and the phenobarbital sodium is freely soluble in water. Third parameter is partition coefficient. It is also an important parameter which affects the biological action of drug molecule. It is defined as the ratio of the concentration of the drug molecule in lipid phase and in water phase. The partition coefficient determines the transport and diffusion of a drug. The diffusion of a drug across the membrane depends upon its solubility in the lipid, that is the partition coefficient of the drug. Fourth one parameter is the hydrogen bonding. Hydrogen bonding is relatively weak bonding which is present between hydrogen atom and an electronegative atoms like fluorine, chlorine, nitrogen, oxygen and sulfur. It plays an important role while combining with the receptor sites and help in stabilizing the structure of a drug. Fifth parameter, that is a protein binding. There are the two types of protein binding with the drugs. First is the intracellular protein binding and the second is the extracellular protein binding. These protein binding of the drugs helps in absorption, solubility and the distribution of the drugs throughout the body, but it does not cross the blood-brain barrier. Sixth point is the chelation parameter. The chelates, these are formed by the metal ion and electron donating molecule. Chelation is an important property which affects the biological action of the various drugs. For example, disodium EDTA, it is the most common chelating agent. Seventh parameter, that is the bioisosterism. Certain drugs, they have the same physical and chemical properties and they produces the similar biological properties are known as bioisoters, and the relationship between the bioisoters is known as bioisosterism. So bioisosterism is of great importance in the drug design. In order to increase the biological or physical property of a drug without changing its chemical structure, bioisosterism is required. It is also used to reduce the toxicity, to alter the activity or to change the bioactivity or bioavailability of a drug molecule. Eighth parameter, that is the optical and geometrical isomerism. So first we should know the stereochemistry. The stereochemistry of a drug molecule, it greatly affects the action of the drug. Isomerism, that the molecules that have the same number of the same kind of atoms but they are having the different physical and chemical properties, that is known as the isomerism. And regarding the optical and geometrical isomerism, there is only one difference, that the optical isomers having the pairs of the compounds which appears as the mirror image of each other, whereas geometrical isomers are also having the pair of the compounds containing the same substituents attached to the carbon-carbon double bond but differently. That is the main difference. So these are the some 8 parameters that affects. So thank you everyone.